So what are the components of blood? As we mentioned earlier that blood is the only fluid connective tissue that is made up of cells, fragments of cells, and dissolved biochemicals. By analyzing the components of blood, one can gain clues about diseases and disorders throughout the body, which is why blood tests are performed more often than any other medical test. Interpreting those results, though, depends on a solid understanding of the components and functions of blood. So let's look at its composition that makes it a vital fluid for life. Blood is composed of formed elements that make up 45% of blood, and plasma that makes up remaining 55% of blood. Formed elements of blood are actually the cells and cell fragments found in blood, such as erythrocytes or red blood cells, 95%, leukocytes or white blood cells, 0.1%, and thrombocytes or platelets that make up 4.8% of formed elements. Of these, only the WBCs are complete cells containing nuclei and organelles. Most of the formed elements exist in the bloodstream for only a few hours or days before they are replaced by new cells. Other than these formed elements, the liquid portion of blood is called plasma, and it accounts for 55% of blood. This plasma is a combination of 92% of water, 7% plasma proteins, and approximately 1% of other substances that we will discuss later under plasma. The combination of plasma and formed elements is called whole blood. In a lab, when a blood sample is spun down in a centrifuge, the heavier formed elements, that are erythrocytes, sink and form a layer at the bottom of the sample, whereas the plasma rises at the top. And a thin whitish-colored layer called the Buffy coat is present at the point where the RBCs join the plasma. This layer is made up of leukocytes and platelets. So these are the main components of blood. Let's learn about their physiology in detail. Starting with the liquid portion of blood, that is the plasma. Plasma is a clear, straw-colored liquid made up of 90% water with organic and inorganic biochemicals. Plasma helps to transport gases, hormones, nutrients, and vitamins throughout the body. And it also regulates fluid and electrolyte balance, as well as pH levels. Normally, it is a straw-colored or pale white liquid, but under certain conditions, a change in its color indicates problems taking place inside the body. Pink-colored plasma suggests the presence of hemoglobin caused by hemolysis or lysis of RBCs, and so the release of hemoglobin that is the component of RBCs into the plasma. A brown or green color may reflect elevated bilirubin levels. Plasma can also be cloudy in cryoglobulinemias. That is the presence of abnormal clumps of proteins named cryoglobulins in blood. Now let's look at detailed composition of plasma. In many respects, the composition of plasma resembles interstitial fluid. That is the fluid between intercellular spaces. Only slight difference comes on account of volume occupied by proteins and their electrical charge. Blood plasma is composed of water that provides plasma approximately 90% of its volume and acts as dissolving and suspending medium for blood solutes. Then plasma proteins, the second main component, that are mainly responsible for maintaining the osmotic pressure of blood and perform many other functions. Electrolytes, that can be cationic or positively charged, that include calcium, magnesium, potassium, and sodium, and anionic or negatively charged, that include bicarbonate, chloride, phosphate, and sulfate. Other than these, plasma also contains some nitrogenous wastes that are the byproduct of various cellular processes. Mainly include ammonium salts, creatinine, urea, and uric acid. Some organic nutrients, that are normally absorbed from digestive tract and are transported by blood to be used throughout the body. It includes amino acids from protein digestion, simple carbohydrates such as glucose, cholesterol, fatty acids, fat digestion products such as glycerol and triglycerides, and vitamins. And lastly, plasma contains some respiratory gases. That includes carbon dioxide, transported dissolved as bicarbonate ions or bound to hemoglobin, in erythrocytes, and oxygen, primarily bound to hemoglobin inside erythrocytes. Among these components of plasma, water makes up around 92%, and 7% contribution is of plasma proteins. So we can say these proteins in water make up around 99% of plasma. The rest of the components are present, but in very less amount compared to these two. So here we will only talk about one of the major components of plasma, which is plasma proteins. The plasma proteins are the most abundant of the plasma solutes, making up approximately 7 to 8% of plasma weight. The liver synthesizes and releases more than 90% of the plasma proteins. So we can say the origin of all these plasma proteins is liver. The three principal plasma proteins are albumins, fibrinogen, and most globulins. All these proteins mainly function to control oncotic pressure, transport substances, hemoglobin, lipids, calcium, and promote inflammation and the complement cascade. Let's discuss these plasma proteins and their functions in detail one by one. Starting with albumin. Albumins are the smallest of the plasma proteins, but make up around 60% of these plasma proteins by weight. The plasma concentration of albumin ranges from 3.5 to 5.5 gram per deciliter, providing the body with a total plasma albumin pool of approximately 135 grams. We know that all plasma proteins are synthesized by liver. So albumin is also made in the liver at a rate of approximately 120 milligram per kg body weight. And because of catabolism, has a half-life in the circulation of almost 20 days. Now, because albumin is synthesized in liver, so any disease in liver will ultimately affect the quantity and thus the performance of albumin, such as plasma concentration of albumin is typically decreased in hepatic cirrhosis, as any damage to liver can cause decrease in albumin concentrations. On the other hand, the hepatic synthesis of albumin is strongly enhanced by low plasma colloid osmotic pressure. Because to maintain it, blood needs albumin. Let's see what are the roles of albumin. Albumin play an important role in the transportation of certain ligands. These ligands can be endogenous, such as bilirubin, ions, hormones, fatty acids, etc. And they can be exogenous, such as drugs. For example, propranolol, methotrexate, etc. Severe liver diseases can cause hypoalbuminemia. In that condition, there will be fewer binding sites for exogenous drugs. And as a result, large amounts of unbound drug exists in blood, leading to increased drug sensitivity. Albumin are also responsible for regulating osmotic pressure inside the blood vessels. Osmotic pressure is the minimum pressure that needs to be applied on solution to prevent fluid movement across a semipermeable membrane. Here, capillary walls are semi-permeable membranes, and plasma proteins, due to their large size, apply that required pressure on blood. Also, albumin has negative charge. With it, they attract positively charged molecules and ultimately creates an osmotic pressure to hold water in the capillaries, which is also known as colloid osmotic pressure. This helps regulate water movement between blood and tissues to aid in controlling blood volume and blood pressure. Therefore, albumins act as important blood buffers. Another important plasma protein is fibrinogen, which makes up around 4% of the plasma proteins. Fibrinogen is made in the liver and is the largest in size of the plasma proteins. And it circulates in plasma at concentrations of 150 to 300 milligram per deciliter. This concentration of fibrinogen gets affected by certain conditions, like the acute phase response, that are the series of events that occurs in response to infection, greatly enhances fibrinogen synthesis. While in case of blood loss or traumatic injuries or liver diseases, fibrinogen decreases drastically. Let's look at its role. Fibrinogen is the major plasma protein involved in blood coagulation. Different clotting factors are also involved in blood coagulation through coagulation cascades, the endpoint of which is the cleavage of fibrinogen into fibrin monomers that further assemble into a large, insoluble strand of fibrin polymer. This substance provides the basic framework for a blood clot. As during clotting, the cross-linked polymers of fibrin form strands that trap red and white cells, platelets, and plasma inside the thrombus, that is, blood clot. Then subsequent shrinkage of platelet plug takes place to ensure uninterrupted blood flow inside vessels. In this way, this fibrinogen together with other coagulation factors play a role in blood clotting. Now lastly, the subtraction of the albumin and fibrinogen moiety from total protein concentration yields the concentration of all the proteins grouped as globulins. These globulins make up around 36% of the plasma proteins. These globulins that are found in plasma are of more than one type. To fractionate these plasma proteins, a technique, namely electrophoresis, can be used. In this technique, plasma proteins move or cover some distance under an electric field. This electrophoretic mobility of a protein depends on its molecular weight, that is, size and shape, as well as its electrical charge. This electrophoresis is usually done on blood serum that lacks fibrinogen and clotting factors. And as a result, a graph is obtained, where two abundant peaks, one of albumin and the other one of gamma globulins, are observed, while two slightly minor peaks of alpha and beta globulins are also observed. So we can say there are three types of globulins in blood plasma: alpha, beta, and gamma globulins. The normal concentration of alpha globulin is 0.1 to 1.1 gram per deciliter. Beta globulin is 0.5 to 1.2 gram per deciliter. And of gamma globulin is 0.7 to 1.7 gram per deciliter. The alpha and beta globulins produced by the liver are mostly transport proteins that bind to fat-soluble vitamins, lipids, and metal ions and transport them to their target site. Whereas the gamma globulins include the immunoglobulins or antibodies that can be subdivided into Ig, IgM, IgA, IgD, and IgE. These immunoglobulins are synthesized by B lymphocytes or plasma cells. The main role of these gamma globulins are in adaptive immunity, where they recognize foreign invader and thus initiates appropriate effector mechanism against them for the eradication of that foreign invader or antigen. So this was all about major proteins present in blood, that are albumins, fibrinogen, and globulins. Other than these three, some other proteins are also present in plasma, but in negligible amount. After these components of plasma, now let's look at the functions and clinical significance of plasma. As plasma forms the liquid base of blood, the functions carried out by plasma and blood overlap, so it performs same functions as of blood. Let's revise them briefly. It plays a role in coagulation due to the presence of fibrinogen and coagulation factors. In defense, that is by immunoglobulins or antibodies, synthesized by plasma cells. in the maintenance of osmotic pressure around 25 millimeters of mercury by plasma proteins like albumin, in the transport of respiratory gases, where it carries oxygen from the lungs to all body cells and cellular carbon dioxide from cells to the lungs, in the transport of nutrients like glucose, lipids, and vitamins absorbed from GIT, transported to all body parts, where they are used as fuel for cells. It also plays a role in the excretion of nitrogenous waste produced as a result of cellular products by some excretory organ. It transports hormones from blood to the target organs. And lastly, it plays a role in the body's temperature regulation by balancing heat loss and heat gain by the body. Other than these functions of plasma, it also has major clinical significance. Where it is used in the form of whole plasma, fresh frozen plasma is indicated in the treatment of massive bleeds resulting in shock, in burns and liver diseases. As in these conditions, the coagulants found in plasma aid in decreasing bleeding time and stabilizes the patient. Fresh frozen plasma also plays an important role as an immediate and effective antidote for warfarin reversal. As warfarin is an anticoagulant, and plasma contains clotting factors to reduce its action. Next clinical use is of its clotting factors. We know that clotting factors play a major role in blood clotting, and these gets activated by any damage in the endothelial cell lining. People with blood clotting deficiencies, such as hemophilia and von Willebrand disease, can suffer massive internal bleeding with a minor injury. Such patients benefit greatly from plasma protein derivatives, such as factor VIII concentrate and factor IX concentrate. Similarly, immunoglobulins from blood plasma can be directly administered under certain immunological disorders like congenital or acquired primary immune deficiency that usually occur when the body cannot produce antibodies or experience the adverse effects of cancer treatments that harm the antibodies. Both disorders benefit greatly from immunoglobulin infusions. Immunoglobulins also play a major role in passive immunization, where it acts as antidotes to diseases such as chickenpox, rabies, hepatitis, etc. Such specific immunoglobulins are derived when patients who have been previously affected by a disease-donate plasma, for example, chickenpox. This plasma contains high amounts of circulating antibodies against chickenpox that can be collected and stored after fractionation for use as post-exposure vaccines for chickenpox. Next use of plasma is in alpha-1 antitrypsin deficiency. It is an inherited disorder that could result in emphysema and cirrhosis in early adulthood. Recent advances in treatment have shown success in decreasing the mortality and frequency of exacerbations when human plasma-derived alpha-1 antitrypsin is given intravenously once a week. Other than this, infusion of albumin, a major plasma protein, is used in the treatment of burns and hemorrhagic shock. Albumin is also useful in the management of the hepatorenal syndrome. Other than these clinical significances, plasma is also involved in many laboratory tests for the diagnosis and confirmation of diseases like diabetes, etc. In this way, plasma performs many important functions, not only under physiological conditions, but also it contributes to treatment under pathological conditions. So this was all about the first component of blood, that is plasma, where we discussed about its composition, plasma proteins, functions, and clinical significance of plasma. Now let's move forward and learn about other components of blood that are formed elements.